Today we will talk about the translation. Translation simply means the protein synthesis. As we know that transcription means synthesis of RNA and translation means synthesis of proteins. You know that this translation, what is simply called as protein synthesis, it is a very complex and elaborate process, and we need machinery for the translation process to occur. That is important requirements for the translation, or we require what is called as translational machinery. We require ribosomes. These ribosomes, they are composed of ribonucleoproteins and ribosomal RNA. So they are called as ribonucleoprotein particles. These ribosomes, they contain ribosomal RNA and ribosomal proteins. Then we require messenger RNA, what is called as mRNA. We require tRNA, transfer RNA. We require amino acids. Amino acids are the building blocks of the proteins, and aminoacyl tRNA synthetases. These are the enzymes which are required for loading the amino acids on the tRNA. This is what is called as translational machinery. As we know that DNA, how the genetic information flows in an organism. We have got DNA. This DNA, it forms RNA. This RNA can be ribosomal RNA, tRNA, mRNA. This is what is called as transcription. Transcription simply means the synthesis of RNA from the DNA, and then we have translation. We have translation. That means proteins, synthesis of proteins, protein synthesis. But this translation, importantly, it requires what? It requires amino acids. It requires amino acids, plus it requires amino acids, and it requires ribosomes. So this is how genetic information flows in a living organism. So we have got a DNA, it forms RNA, what is called as transcription. This RNA, it gets involved in protein synthesis, what is called as translation. But for the translation process, we require what? Amino acids, which are the building blocks for the proteins, and we require the ribosomes, ribonucleoprotein particles, which are called ribosomes, and these are the factories for the protein synthesis. So what do you mean by translation? How you can define the translation? How you can define the translation? Definition. How you can define the translation? Translation is actually, we have got an mRNA. This mRNA has a particular sequence of nitrogen bases, and this sequence of nitrogen bases determines the sequence of amino acids in a protein. This is what is called as translation. That means the sequence of nitrogen bases in mRNA determines the sequence of amino acids in a protein. That is called as translation. So the sequence of, it is the sequence of nitrogenous bases in the sequence of nitrogen bases in mRNA determines the sequence of amino acids in a protein, determines the sequence of amino acids determines the sequence of amino acids in a protein. This AA represents amino acids in a protein. I will explain it like this. That means we have got proteins. This is protein polypeptide chain. This is polypeptide chain. So this polypeptide chain, suppose this is a polypeptide chain. This polypeptide chain is composed of hundreds of amino acids. We have got amino acid, for example, amino acid 1, amino acid 2, amino acid 3, amino acid 4, amino acid, so on and so forth. That means nth amino acid. So this is a polypeptide chain. These are simply the amino acids. AA represents what? Amino acid. A is amino acids. This AA represents the amino acid. So we have got amino acid 1, amino acid 2, amino acid 3, amino acid so 4, so on and so forth in a polypeptide chain. And these amino acids, the adjacent amino acids in a polypeptide chain, are joined or linked by means of a bond that is called as peptide bond. This peptide bond, it is present between the adjacent amino acids in a protein. Now I told you that what is translation? Simply we say translation is the protein synthesis. Now how we can define the translation? It is simply the sequence of nitrogen bases which determine the sequence of amino acids in a protein, which determine the sequence of amino acids in a protein. For example, we have got mRNA. We have mRNA. This mRNA, it is having particular sequence of nitrogen bases. Suppose we have, this is the mRNA, this 5' end, this is 3' end. This mRNA, it is having a particular sequence of nitrogen bases. For example, we have AUG, UUU, GUG, UGG, UGG, and we have AAA. So this is a mRNA molecule, messenger RNA. This is a messenger RNA. This is 5' end and 3' end. At the 5' end we have a phosphate group free, and at the 3' end we have a hydroxyl group free. 5' represents the carbon at the 5th position, and 3' represents 3' represents the carbon at the position 3rd in a sugar. So this is a particular sequence of nitrogen bases. This is a particular sequence of nitrogen bases on mRNA. This AUG code is for now polypeptide chain. Polypeptide chain. This polypeptide chain would be AG codes for what? It codes for methionine. So that means the sequence of nitrogen bases in mRNA, it determines the sequence, which type of sequence would be present in the polypeptide chain. So obviously we know that AUG codes for methionine, UU codes for phenylalanine, and GUG codes for valine, and UGG codes for tryptophan, and AA codes for lysine. So this is the polypeptide chain, and these are the amino acids. This is the sequence of nitrogen bases. We can write it is a sequence of nitrogen bases. Sequence of nitrogen bases. And this is a polypeptide chain, and there would be the sequence, particular sequence of amino acids. So this is sequence of amino acids. So how we can define a translation is simply the sequence of nitrogen bases. This is the sequence of nitrogen bases in mRNA. This is the sequence of nitrogen bases mRNA, and it determines which type of amino acids, what would be the sequence of amino acids in a protein. The sequence of nitrogen bases in mRNA determines the sequence of amino acids in a protein. This is what is known as translation. So translation simply means the protein synthesis, and it can be defined as the sequence of nitrogen bases in mRNA which determine the sequence of amino acids in a protein. And then we have got, this was regarding the definition of the translation. Then what is the mechanism of translation? So before that, it is necessary that what are the requirements for the translation process. What we require ribosomes. Ribosomes are simply what is called as translational machinery. It is what is called as translational machinery. We require ribosomes, which are called as protein factories, mRNA, messenger RNA, because the information is coded in the messenger RNA. We need transfer RNA, which is responsible for transfer of amino acids from the amino acid pool to the ribosomes for the protein synthesis to take place. And then we require amino acids, as we know that the amino acids are the building blocks for the proteins. Then we require aminoacyl tRNA synthetase enzymes. We know that these amino acids, they are needed to be loaded on the tRNAs, and this activity is being performed by the enzymes, aminoacyl tRNA synthetases. There are separate aminoacyl tRNA synthetases for these amino acids, which get loaded on the transfer RNA to get transported from the amino acid pool to the ribosomes for the protein synthesis. So we'll discuss about the mechanism of translation. This mechanism of translation, how this translation takes place, we'll discuss its mechanism. The mechanism of translation. First step, it is basically a multi-step process. This translation, it is a multi-step process. First step is charging of tRNA. First step is activation of amino acids. Then we have got second, charging of tRNA. Third is initiation of protein synthesis. Then elongation of polypeptide chain, and third is termination of protein synthesis. So mechanism of translation. Mechanism of translation. Mechanism of translation. First we have activation of amino acids. Activation of amino acids. Mechanism of translation. The first, it is a multi-step process, and the first step is activation of amino acids. Then we have got charging of tRNA. Charging of tRNA. Then initiation of protein synthesis. Initiation of protein synthesis. Initiation of protein synthesis. Initiation of protein synthesis. Then we have another step, that is elongation of polypeptide chain. We have elongation of polypeptide chain. Elongation of polypeptide chain. Elongation of polypeptide chain. And last step, that is termination. Termination of protein synthesis. termination of protein synthesis. Termination of protein synthesis. First we will take activation of amino acids. Activation of amino acids. Activation of amino acids. Activation of amino acids. These amino acids, I told you that these are the building blocks. Amino acids are the building blocks for the proteins, and there are many amino acids which are present in a cell. There are actually 20 amino acids participate in the protein synthesis, but in a cell more than 20 amino acids can be present. This their activation is important, as we have got these amino acids plus ATP these are called activated amino acids. Activated amino acids. This amino acids plus ATP, and this activation process done by the enzyme that is called as aminoacyl tRNA synthetase enzymes. Aminoacyl tRNA synthetase enzymes. So we have got amino acids. They are activated by their reaction with the energy in the form of ATP. This reaction is catalyzed by the enzyme aminoacyl tRNA synthetase enzyme, and it gives what is called as activated amino acid. Then we have got another step that is called as charging of tRNA. Charging of tRNA. This tRNA, it is responsible for the transportation of activated amino acids from the amino acid pool to the ribosomes for the protein synthesis to take place. So we have got, you know that we have got tRNA. It has got a cloverleaf model of tRNA. We have got this structure of tRNA. This is called as 3' hydroxyl end, and obviously this is 5' phosphate end of tRNA, and this 3' hydroxyl end of tRNA molecule, this is a tRNA molecule. It is the amino acid binding site. This is amino acid binding site. That means at this site amino acid gets bound. For example, this is amino acid. This is what is called as amino acid binding site. This is amino acid, and this is called as amino acid binding site. Then we have got another. This is called as T-C loop. This is ribosome binding site. Ribosome binding site. This is what is called as anticodon loop. And we have got here that is called a dihydrouridine loop, DHU loop. This dihydrouridine loop, it is the aminoacyl tRNA synthetase enzyme binding site. It is an aminoacyl tRNA synthetase binding site. It is an aminoacyl tRNA synthetase enzyme binding site. This in case of tRNA, this 3' hydroxyl end, it extends beyond this 5' phosphate end. So I have made it a this line is a little bit stretched as compared to that of this line, because in tRNA the 3' hydroxyl end is extended as compared to that of the 5' phosphate end, and this 5' phosphate end, you know that it carries 7-methylguanosine. It carries 7-methylguanosine cap. 7-methylguanosine cap, this 5' end. This amino acid gets, this is the amino acid binding site. Amino acid gets attached to the tRNA at the 3' hydroxyl end. Then this amino acid is being carried to the ribosomes, and this loading of this amino acid to the tRNA is being performed by aminoacyl tRNA synthetase enzyme. This is the charging. This is what is called as charged tRNA. That means amino acid present at 3' hydroxyl end, and what is called as charged. This is what is charged tRNA. This is what is charged tRNA. Then we have got another step, that is initiation of protein synthesis. Third step is, these are two important steps, that is activation of amino acids, where amino acids are activated by their reaction with the ATP. This reaction is catalyzed by the enzyme, aminoacyl tRNA synthetase enzyme. It gives activated amino acid, and then we have charging of tRNA, where this activated amino acid is loaded on the tRNA at the 3' hydroxyl end, and it gives the what? Charged tRNA. Then we have got an important step, that is initiation of protein synthesis. This is initiation of protein synthesis. Initiation of protein synthesis. Initiation of protein synthesis. We know that ribosomes are simply called as protein synthesizing machines. These are protein factories. Ribosomes are simply protein factories, and we have got what? You will see a ribosome. It has got two subunits. This ribosome, it has got how many subunits? It has got two subunits. This is small ribosomal subunit. This is small ribosomal subunit, and this is larger ribosomal subunit. Obviously, this mRNA chain, this is present here. mRNA chain is present here. This is mRNA, 5' end, 3' end. This is messenger RNA. This is messenger RNA. And in the ribosome, I told you that these are the protein synthesizing machines, and there are 3 sites in this ribosome. One is called as, this is A site, P site, and E site. This is messenger RNA. These are 2 subunits of ribosomes. This is A site, P site, and E site. This A site this is P site, and this is E site. This E site, what do you mean by A site? This is A site, P site, and E site. A site is acceptor site, amino acid acceptor site. P site is peptidyl site, and E is exit site. So A is aminoacyl acceptor site, P is peptidyl site, and E is exit site. These are the 3 important sites in the ribosomes. One is A site, P site, and E site. A means aminoacyl acceptor site, P is peptidyl site, and E is exit site.